Good morning and welcome. Very happy to see you here bright and early. Thank you for joining us. My name is Stefano Bertuzzi, and I am the Chief Executive Officer of the American Society for Microbiology, ASM. I'm glad you could come today for the rollout of the report, Application of Clinical Microbial Next Generation Sequencing, and you should have received a copy of the report. We have a very exciting program today, which is made possible by our sponsors, and they are here on the panel with us, and they are bioMérieux and Illumina. And we really thank them for their support for this initiative. Without them, it would have not been possible for sure. So thank you very much. A round of applause for our sponsors. Microbial sciences are undergoing a phenomenal renaissance, which is triggered in large part, at least in my view, by the very powerful new technologies. And nothing better than next generation sequencing exemplifies this fact in my mind. With only 10% of the microbes in our human bodies which are culturable, we have been, of course, very, very limited in what we can learn about microbes. The landscape totally changed with the advent of new sequencing technologies, which allowed us to look under the hood of the human machine and discover the marvels and also the threats coming from life's most powerful engines, which are the more than 100 trillion microbes which live with us and which greatly outnumber our own human cells. Since we are in Washington, a way of thinking about this is that if our bodies were to take a vote, we humans would really lose. And this has been possible really through the advent of this technology, which changed the possibilities for the field. Next generation sequencing has already changed the landscape of basic science and basic microbial sciences in particular. And we're now poised to see what could be a radical change in clinical microbiology as well. Although this has not yet happened for multiple reasons, which is the reason why we're here today. In fact, as of today, while we have, for example, FDA-approved test systems which allow laboratories to develop and validate sequencing of any part of the patient genomes, or assays to detect changes in cystic fibrosis transmembrane conductance regulator, CTFR, we do not yet have FDA approval for microbial diagnostic use. So that is a bottleneck, and that is something that we want to talk about today. With next generation sequences, there are enormous opportunities in front of us for clinical microbiology, from rapid and accurate diagnostic for known and, very importantly, for unknown pathogens, which the unbiased systems allow us to tackle in a more efficient way. Think about precision medicine. Think about one of the biggest challenges in front of us, which is the antimicrobial stewardship. And I can keep going on with the list of opportunities in front of us. But there are also barriers for next generation sequences in clinical microbiology and in public health laboratories, for which we need to lower the activation energy. And these barriers are things like the cost of entry, how expensive the technologies are for a lab to be able to afford it at a standard level. The extremely complex data analysis and the reporting, the lack of standardized reporting formats, and as a consequence, the lack of regulatory guidelines on this front. So this is why last April, the Academy of Microbiology gathered a group of clinicians, of researchers, government officials, industry professionals together to answer a series of questions about the role of next-generation sequencing in the clinical microbiology laboratory. ASM is positioned to advance the microbial sciences by leveraging the power of our members. We have 47,000 of them, with an amazing brain thrust, which is in the Academy of the American Academy of Microbiology. And I would like to recognize Dr. Michelle Swanson, who's here in the audience, who's the chair of the board of governors of the Academy, and also Marina Moses, who's the director of the Academy. And I don't see Marina, but she's somewhere in there. She's right in front of me, yes. The American Academy of Microbiology is an honorific leadership group of ASM with the mission to recognize scientific excellence as well as foster knowledge and understanding in the microbial sciences. Next-generation sequencing is a tool that has boundless ways to help so many industries. For one, the food safety industry by quickly detecting pathogen that could cause contamination and trigger outbreaks. Moreover, with next-generation sequence's ability to detect mutation causing pesticide resistance, the agricultural field can greatly benefit by protecting crops and better improving plant health. So the sky is the limit for the application of these technologies. And so the list of potential benefits are endless. And I'm really excited today that we have with us a distinguished panel of speakers that will help us focus the topic on the clinical microbiology aspects. So our panelists are here at my left, Dr. Charles Chiu, who's associate professor at the University of California, San Francisco, who decided to abandon the fog for the beautiful Washington weather. And Dr. Chiu is one of the first physicians to use next-generation sequencing to detect and cure an infectious disease, neuroleptospirosis, which was published in a landmark paper in the New England Journal of Medicine. We wish it was in an ASM publication, by the way. Think about it next time. He was also a participant in the application of clinical microbial next-generation sequencing Colloquium. And Dr. Chiu will talk about the opportunities of using NGS in diagnostics. So thank you for being here today. To his left is Dr. George Weinstock, who's the chair of the Colloquium Committee and faculty at the Jackson Laboratory for Genomic Medicine. Dr. Weinstock will provide an overview of the Colloquium report and the impact that we think will have on the field. Thank you, George, for being here today. To George's left is Dr. Joseph Campos. Joe is the ASM secretary and devotes a very large amount of his abundant spare time to ASM. During the daytime, he is the interim chief of the Laboratory of Medicine at Children's National Health System here in Washington. ASM has not been standing still since the Colloquium. This is fast-moving topic. And Joe will talk to us about what we're exploring for the future on this front and how we will move forward after today. In addition, we have, to the speakers, we have two panelists, which I mentioned before, from our sponsor. We have Dr. Mike Dunn at the very end of the table from bioMérieux, and I hope you appreciate how I say it, you know, in the French way. Who is Executive Director of Research and Development in North America, and Ms. Kelly Hoon, who's the Commercial Development Manager at another landmark industry in the field, Illumina. So without further ado, thank you very much for being here, and we can start from you, George. Charles.
Good morning. So I only have 15 minutes, and I just want to start out by saying that clinical microbiology and its applications, next-generation sequencing applications, is a very broad field. I'll be focused on rather two very narrow aspects of it, but I just want to show you this to give you a glimpse of the potential of metagenomic next-generation sequencing or next-generation sequencing applications in general for diagnosis of infectious diseases. And the goal of what we're doing at University of California, San Francisco is we want to implement this as a way to impact clinical decision-making. And the goal is what we call precision diagnosis. It's under a moniker of precision medicine. And the goal of that is, can we actually implement the latest technologies to generate data that can make an immediate impact on patients? This has already been shown to be efficacious in many different fields, including cancer, prenatal testing, but really for microbiology and infectious diseases, this is an area that's remained largely unexplored. And it's our goal to apply a next-generation sequencing test, what we call metagenomic next-generation sequencing, to be able to diagnose unknown infectious diseases in patients as a way to provide actionable information that can lower costs and improve patient outcomes. So our goal is to target infectious diseases of clinical unmet need. And this includes pneumonia, for up to 25% of the time, patients in the intensive care unit don't get a diagnosis, despite extensive testing. For diseases such as encephalitis and meningitis, the cause can be up to 70% of the time, the cause remains unknown. And even for other infections that are well characterized, like infectious disease syndromes like acute hemorrhagic fever, 20 to 30% of the time, the cause remains unknown. And using these new technologies, what we can do is, in principle, there's no reason why a single test that's based on sequencing can diagnose the full spectrum of pathogens that cause infectious diseases. So we can, with a single test, in principle, determine whether or not your disease is due to a bacteria, a virus, a fungus, or a parasite, the full spectrum of microbes that cause infectious diseases in humans. So the general idea is called metagenomic sequencing. What we're doing is we're simply taking clinical samples, whether it's blood, cerebrospinal fluid, nasal swabs, stool, etc. And the goal is to cast a wide net and to analyze those samples and be able to determine what organism may be present and causing disease in those samples. So we're using a huge fish net, as you can see here, instead of a single fishing pole. And this has been made possible over the past several years by the vast improvements in sequencing technology. And what you see on the right is an Illumina HiSeq instrument. And this is an instrument that can produce up to 1 billion sequences per run. And the goal of this is that we're taking an unbiased approach, and we're simply sequencing everything in your sample and be able to identify what virus, bacteria, fungal, or parasite may cause disease. The other thing that's also been, in terms of development, that has kind of paralleled the development of sequencing technology has been the ability to analyze that data. And this really gets into where medicine meets kind of big data. And the goal of this is that we've developed a pipeline called the SERPI bioinformatics pipeline, but many different groups have developed pipelines that can analyze millions and millions of sequences. And ultimately, the goal of many of these pipelines is to be able to implement them on the cloud, so you can take advantage of cloud computing, and that you can actually analyze your data in a turnaround time that's clinically actionable. For infectious diseases, we don't have days to weeks, we actually have hours to days, really, to be able to make a diagnosis, especially for critically ill patients. In addition, the other key component here is that we need to develop this in the context of a CLIA laboratory, a clinically licensed laboratory. And this is for many reasons. For all clinical diagnostics, they have to be done under clinically licensed laboratories that are under federal regulation. And it's what you see here is an example of a typical sequencing clinical workflow where you go directly from your sample to ultimately to the result, to where you can provide information that can be usable by a clinician. So a doctor who does not have bioinformatics expertise may be able to analyze that data and may be able to look at those results and leverage that data to take care of his or her patients. This gives you one example of the kind of power of this approach. So what you actually are seeing here are basically 6 different individuals with various fever, febrile, and respiratory illnesses. And I just use this as an example to show you that we can diagnose the full spectrum of infectious diseases. You can see here that we can diagnose 2 types of viruses, and you see on the top row. We can identify bacterial infections, Haemophilus influenza, typhoid fever, even a parasitic infection, which is malaria, which is caused by a parasite called Plasmodium falciparum. And what this really does is it suggests that this sort of breaks the paradigm of one test, one bug. Typically, infectious diseases, what we do is that for diagnosing influenza, for instance, a clinician will order an influenza test. If the clinician thinks that it's Ebola virus infection, that clinician will order an Ebola test. This subverts the paradigm, because what we can do is we can have a single test that will, in principle, detect everything. What you see here is Josh Osborne. He's a 14-year-old boy, and his family and he have consented for me to share his story. He is a boy who went to a trip to Puerto Rico and came back with kind of fever and a rash and had a flu-like illness. Ultimately, over the next 4 months, he underwent 3 hospitalizations, spent 44 days in the ICU, had over 100 inconclusive tests, underwent 3 empiric treatments, including empiric antibiotics with no effect. and ultimately became critically ill because he went from getting a flu-like illness to developing essentially a brain infection, encephalitis and meningitis, neck stiffness, was in an intensive care unit and underwent a brain biopsy where they take a piece of tissue from his brain to see whether to try to diagnose his presumed infection. Ultimately became critically ill and had to be put in a coma, medically induced coma for having seizures that were uncontrollable. And it was around the time that he became critically ill, and this was 4 months after he initially presented with symptoms to the emergency department. We received a sample on a Friday. We were able to run the next-generation sequencing test, and we actually got a result in 48 hours, and it was unambiguous. This was actually an undiagnosed case of neuroleptospirosis. And ironically, the treatment of choice for neuroleptospirosis is our oldest antibiotic, which is intravenous penicillin. And it turned out that after we switched him to penicillin, he was out of the ICU and cured within 2 weeks after being given the appropriate treatment. At the time, we didn't really have any kind of automated visual interface at the time, so we had to analyze the data manually. But what you see now is that we've been developing a visual interface. So you can see here, this is what you actually see on the left corner is on the right corner is a heat map. On the left column is actually DNA, which is directly extracted from his cerebral spinal fluid, from his spinal fluid. And you can see here at a glance that this was a case of neuroleptospirosis from Leptospira santarasi. What's interesting is that using sequencing, we were able to identify the exact species. Santarasi is actually only found in 3 places in the world, or has been found in 3 places in the world, including Puerto Rico and Taiwan. Before, what we had to do is actually we had needed to have a bioinformaticist take a look at all of those millions of sequences, run the programs, analyze the data. Now, at a glance, the goal is that a clinical technician or a clinician could even look at the results and be able to obtain a diagnosis. And I just want to end by talking about a few things. One is that we're working on a project called the California Initiative to Advance Precision Medicine. And the goal of this is a project which has been funded by University of California and the California state government. This is a coalition of many different University of California institutions you see on the top, as well as collaborations with many institutions below. And our goal here is we plan to actually enroll 300 patients from these 3 UC institutions, run the test in a clinically licensed CLIA laboratory, be able to analyze the data on the cloud, and to have the results seamlessly integrated into patient electronic medical records. And it's our goal that this would be a demonstration project over the next year that would set the stage for future deployment of this technology in clinical laboratories. And for those of you who are skeptics who may say, well, when are you actually going to get this done? I actually here have taken the put up a dangerous slide saying that we actually have a timeline. And the goal here is that we are actually planning to launch a clinically licensed laboratory developed test in May of this year, and we're actually hoping to actually launch the prospective study in May as well. So this will be a 1-year study, and we have a very aggressive timeline, as you can see here. I want to end because I want to talk about other technologies as well. We're working on this other technology called nanopore sequencing, and this is probably a very recent sequencing technology. And what nanopore sequencers are these very small devices. You can see here that they're slightly larger than a USB stick. And in fact, these devices are actually powered by a USB laptop. And the goal here is that we can actually do the sequencing kind of in point-of-care settings. And we've been able to use it for real-time pathogen identification. One application, as you might expect, would be in addressing febrile outbreaks, or really in point-of-care settings. And you can see here, this is where we can actually identify viral reads. These are from patients, blood samples from patients infected with Ebola virus and chikungunya virus. And you can see that we can identify them within 10 minutes of starting the sequencing. We've been able to use this to show that we can, for instance, use this as a technology to track the emergence of chikungunya virus, which is a mosquito-borne virus that causes a febrile illness in Puerto Rico, using nanopore sequencing. I just want to start by showing you a short video of kind of the speed of this technique. So what you're seeing here is we're using kind of the similar pipeline that we have, and I've run this kind of 8 times fast forward. And the goal is that once we start the sequencing, you can see within about a minute, you start to actually upload the sequence data, and then you get kind of the sequence calls that come back within 2 or 3 minutes. Ultimately, you start to actually have an identification in there of human reads, and then if you look for another 30 seconds, we can identify Ebola virus. And what's important about this is that we're not looking to actually we're not using a test specifically for Ebola virus, we're just using a test for anything. It could have come up chikungunya virus, it could have come up Zika virus, it could have come up really MERS coronavirus. And that's the goal, is that we're not targeting any one pathogen, but really looking for everything simultaneously. This gives you some example of the potential of this technique. We're actually using this now to do Zika virus testing with numerous collaborators in South and Central America and the Caribbean. And this is some very early data which we just generated this week, which basically suggests by using sequencing, for instance, we can achieve essentially equivalent sensitivity to using a very specific test called polymerase chain reaction for Zika virus detection. But the advantage of the sequencing approach is that we're not targeting Zika virus. In fact, we actually noted that one of the samples that we were screening for Zika virus was actually a patient who was unfortunately co-infected with 2 viruses, Zika virus and chikungunya virus. So I just want to summarize with 3 short points. One is I think that emerging genomic technologies are very promising for what we call precision diagnosis. New bioinformatic pipelines such as SERPI, which was developed here at UCSF, greatly improve computational turnaround times and make this data accessible to clinical and public health laboratories. And then finally, there are many regulatory and technical challenges, but I feel that next-generation sequencing technologies have evolved to where we can now implement them clinically. And I don't think the time frame is really 5 to 10 years. I'm actually hoping that the time frame is more 1 to 5 years. Thank you very much.
Questions at the end.
Hello, everybody. I'm George Weinstock. It's great to see all of you here to talk about this report. So this is the report on the applications of clinical microbial next-generation sequencing. What does all that mean, and why did we want to do that? So next-generation sequencing is 10 years old. And really, those first couple of years or so were a lot of instruments that didn't work very well, and it was still being developed. So it's really just a handful of years old that this has hit. And we're really talking about something that's a state-of-the-art technology. But everybody's using it. It's driving cancer genomics. It's driving many things in microbiology. It's just transforming many, many different areas of medicine. All the personalized medicine, precision medicine, obviously medicine is personalized and precision for a long time. So why is there all the fanfare about it now? Because of next generation sequencing, because your genomes are being sequenced or your microbiomes are being sequenced, and there's this new kind of genetic-based information that is just changing things and making it much more immediate and much more tailored to the patient. So the impact of next-generation sequencing is just enormous. And like any new field, everybody who's using it, after a while starts to try to think of, how are we going to be more systematic? How are we going to pull this together and develop an industry and to develop products? But of course, there's not like an association for next-generation sequencing users or something like that. It's just distributed throughout the research community and throughout industry and so forth. So what became apparent to people as it was so rapidly starting to have impact, like the story that Charles just told about the leptospirosis patient, is that we really need to get going on this. We really need to try to figure out how to penetrate this into the clinical space so that it can be used. Companies like Illumina and bioMérieux have been helping to foster this, supporting meetings, putting together interesting programs to do that. But really, I think what was great about this, and in my history with the ASM novel, is that the American Society for Microbiology stepped up and took ownership of this issue and used the vehicle of the American Academy of Microbiology in order to do that. And this is sort of, labels the American Academy of Microbiology as kind of trying to be ahead of the curve and visionary and things like that, and take ownership of an area that is really crying out to have somebody do something with. And that was really the driver behind a lot of this. And so the result is this wonderful report, over 50 pages long, a lot of reading to do here, a lot of good pictures in it though, yeah. And I think that this is somewhat unique in that it attempts to take a huge heterogeneous field that's rapidly evolving, and you can be guaranteed that 2 years from now the instruments and the technology and the software and the applications will be things we can't imagine right now, because that's the way it's been in genomics since the Human Genome Project started. But nevertheless, to try to make sense out of that, make coherence out of that, and make some recommendations and get people's thinking focused on the things to do to take advantage of the technology as it continues to unfold. So it's really a tremendous success, I think, and very much to the credit of the ASM for doing that. So here was our committee. Besides myself, Brittany, Nate, and Heike were absolutely key in running this process that has had many moving parts to it. And here were the members of the Colloquium. The Colloquium was the mechanism that the American Academy for Microbiology decided would be, let's assemble some people who represent basic research, clinicians, regulatory agencies, industry, just try to cut a broad swath of perspectives and lock them in a room together for a couple of days and have them think this through. And let's come up with this report, as I said, that tries to guide thinking about how we're going to develop this. Charles was on this, Kelly was on it, Mike was on it. Mike is listed here as William Dunn. He actually has an alias, so we can't always catch up with him. But that's the same guy. And because we had a great organization like the Academy running this, they understand how to, the kind of process that you need to go through to pull something like this together and to make it impactful. There were, a whole series of stages that this went through, defining the questions we were going to do, choosing the right people to be there to address those questions. Then there was the colloquium, then there were draft reports, then there was the final report. This was a lengthy process that was done with the bar set very, very high in terms of what the final product would be, and I think you'll see that that's the case when you look at this report. So one of the first things was to try to figure out what is this in this huge heterogeneous field, what is it we're trying to do? What are the questions we're going to focus on and everything like that? The first one was, what is next generation sequencing good for? And how does it compare with other methods that are out there that are being used in diagnostic labs? Everybody sort of felt that this was an important new technology and that it had these applications, but we really wanted to define that. We wanted to get to specifics about that. The third bullet point, the impact of NextGS if implemented in clinical public health microbiology. What is really going to happen if and when it penetrates into the applied arena? What are we going for here? The top bullet point here, examination of feasibility into workflow, costs, efficiency, things like that. You're talking about changing the way people do things, and the operations that you're talking about affecting diagnostic laboratories and hospitals. These are very impressive operations that are swamped. Lives hang in the balance about their ability to do it, and here next generation sequencing is coming along and saying, oh, and by the way, we want you to do a whole new thing that you haven't done before and you don't know how to do yet, and we think it's going to be useful, but we're not exactly sure how it's going to be useful. So these questions of feasibility and selling it to the hospitals, that this really is important for them, trying to get that defined so that everybody understands what really is the benefit of all of this. Data management issues, you know, we've all heard the term big data. Well, next generation sequencing is, in the context of biology, the poster child for big data. It's not like meteorology or something like that, or Wall Street, where they have really a lot of data. But for biologists, the vast amount of DNA sequence data that's generated by next generation sequencing poses issues of data management and analysis and computational speed and things like that that are new and are still everybody scratching their head about how they're going to solve that, because companies like Kelly's Illumina keep coming up with new instruments that, like I say, 2 years from now, we'll be making, you know, what we produce now in a sequencing instrument, we'll just laugh at 2 years from now, because we'll be making 100 times as much as we do now. So Moore's law and the computational challenges of all of this are in the dust. This is big data, and this poses interesting challenges for clinical institutions. The last 2 bullet points, equally important: deterrents and shortcoming of technology. So we have to not just be Pollyannas, we have to really look hard at this and try to figure out what is the niche that this can apply to, and where is it useful, and what are the problems of getting it implemented, some of which I've alluded to. And then the last one, anytime you have a new technology and you're trying to apply it for life and death matters, you have to worry about standardization, reference databases, things so that everybody's speaking the same language, and standards can be set for that. So you can see this is quite a broad order that this colloquium took on, and all the answers are in here. This is it, you know, don't have to worry about it. There were a number of ancillary activities. This was not a one-off at ASM. They wanted to do a number of other things associated with this. There's a short review article that was published in M.Bio that addresses a lot of these questions. The wonderful conferences program at ASM, which is long-standing and always hits timely issues and brings together smaller groups of people throughout the microbiology community. They had a very timely conference, the ASM conference on rapid NGS bioinformatic pipeline. So this was about this same topic, and this was held in the fall. I've got to say that of all the conferences I've been to, this had a number of unique characteristics to it. This was in Washington. It was like a couple of days, it was the weekend after the Pope visited. Okay, so just sort of remember what was going on in Washington around then. The head of China was coming, and the Chinese embassy, I think, is near where the hotel was. So there were scores of buses, and the streets were lined with lots of Chinese in red garb, just so that when the head of state of China drove past in his limo, he should see tens of thousands of supporters in the area. Now, the police had all the streets cordoned off, and so it was almost impossible to get to the hotel for this conference. Plus, there were all of those. If that wasn't enough, there was an important Republican organization, sort of a thought-leading organization, was having a meeting in the hotel to vet all the candidates at that time. This was before anybody had dropped out, you know, this was early on. And so the hotel was packed with Republicans, and all of the glitterati were there, and all the things. And you know, we had our little conference room and our little hall, but all the other halls had all these booths for all the lobbyists and everybody else that was there. If you just sort of hung out in the lobby, you'd hear these hard-boiled political types talking about, you know, Cruz versus Trump versus this or something. So it was a remarkable combination of things going on with this very timely meeting happening at the same time. I don't think I'll ever quite forget that. And then the Next Generation Sequencing Coalition To be continued. This is the part of this whole process that has legs. ASM wants to continue this discussion. They want to continue to try to figure out how to make sure that the world takes advantage of this technology and gets it applied to infectious diseases and microbiology. And so this will be a new group. A coalition is going to be a group of individuals within ASM who are going to think through what are the next set of activities that ASM should do, maybe evaluate how things are going, stuff like that, but to keep this process going, to keep this topic alive and vital within ASM. Everybody's very interested in it. So again, I think that's another unique step compared to a lot of the things that ASM has done in the past. And I think it's a kind of a tribute to their thinking and wanting to take ownership of this very important topic. Okay, so there are lots of recommendations. I'm not going to go through these in too many details, but I want to just at a high level give you a feeling for what is in the report. Performance factors and infrastructure, development of turnkey solutions, distinctions between clinical and analytical. These are again, by way of defining and starting to say we really need to get some particular procedures and we need to distinguish what those procedures are for. That's very important in terms of reducing this down to a manageable set of topics that regulatory agencies and clinical institutions and the research community can focus on in terms of doing things. Standard operating and validation procedures sort of goes without saying, particularly if you're going to involve regulatory agencies or things. Somebody needs to be thinking about how we're going to deal with these topics. Interdisciplinary teams and education. This has to do with, how do you get something like this to now penetrate into the clinical arena? Next generation sequencing is very new. Sort of nerdy people like me who love sequencing technology and have done genomics for a while just live and breathe this, but a clinician might, know a little bit about it from the New York Times or something like that, and we really need to improve the knowledge, if not the know-how, within diagnostic labs and in the clinical arena so people can start thinking about it more intelligently and try to figure out how their institutions can use this and what needs to be done. Interpretation, sharing, and management of data. So, okay, we're going to generate data, but we need to have common databases. We'd like to have data from clinical situations shared so people can learn from it. There's a whole shtick here, and for basic science genomics, this has been the mantra for a long time, open release of data. When you sequence a new microbial genome or you have metagenomic data or something, you want to share it with everybody so people have data sets they can play with, they can see how you analyze it. Very, very much a transparent field. How to do that in the clinical area is very important too. And then the last one, clinical reporting billing, this has to do with regulatory agencies, insurance companies. Undoubtedly this will become the bottleneck. Many of these other things are more laboratory, researchy, application-based, and we know how to solve those. But there's some new concepts here, and there's some real challenges here, and we want to do this fast so that it's in the hands of clinicians as soon as we can. And so we need to really work arm in arm with regulatory agencies and insurance companies and other people who are really the ones who are going to make this happen in the end and make it appear. So I will stop there. Thank you very much.
Thank you, George.
Well, good morning. It's so exciting to see a large crowd here in this very stately room at the National Press Club to hear about something that's extremely modern and with it today, next generation sequencing. Just to remind you, I'm Joe Campos. I'm the secretary of the American Society for Microbiology, which is an elected position that I've held now for 8.5 years. And with the 9-year term limit that our Constitution and bylaws imposes on this office, I'll soon be stepping down. But I have to say, what a ride it has been, especially in the last few years, as the microbial sciences have leaped forward in the use of technology, what you've heard about earlier today. I thought, since I'm a clinical microbiologist as well, Stefano mentioned that I'm the interim chief of the Division of Laboratory Medicine at Children's National Medical Center here in Washington, D.C., and my entire career has been devoted to pediatric clinical microbiology. And over the years that I've been at Children's Hospital of Philadelphia first, and then I moved down I-95 to Children's National here in Washington, I've been a witness to an amazing succession of technologies. And seated at the front table here with us is Mike Dunn, who has been a lifelong, almost lifelong friend of mine, I think. Mike is a card-carrying clinical microbiologist and a pediatric clinical microbiologist to boot. And many years ago, in the early 80s, we put together a group of pediatric clinical microbiologists who met every year at the ASM general meeting, kind of in an unofficial status. We used to have to hunt for meeting space in the cities where the ASM general meeting was held. But we were able to pull it off and attract a large group of our colleagues who were really interested in pediatric clinical microbiology. And when this meeting began, it was in the days when we were still pretty reliant on culture. Receiving specimens from sick children, inoculating agar plates, and seeing what grew. But over the course of the 25 years or so that Mike and I organized this meeting, we saw the evolution of technology begin with the introduction of rapid antigen detection of specimens, which at the time we thought was hugely exciting. Finally, we had a tool in the microbiology laboratory that was high-tech and could give us an answer in a few minutes as to what organism might be causing a sick child's infection. After that, we saw the beginnings of molecular diagnostics with the invention of the polymerase chain reaction and other technologies that are related, involving amplification of nucleic acids. It wasn't that long until this technology made the leap from the research laboratory into the diagnostic laboratory, and today we are heavily invested in my laboratory at Children's National in the use of the polymerase chain reaction, other amplification technologies, and we're at the threshold of following in Charles's footsteps in launching a microbial genomics laboratory at Children's. So what a time. It's just, I thank my lucky stars that I was a clinical microbiologist when all of this was happening. George did a nice job of summarizing what ASM has been up to in the area of next-generation sequencing. He described the Colloquium, which was a huge success, and I do want to mention the Colloquium was really the brainchild of a few individuals. Of course, Marina Moses, who you've already met, was very involved, and Michelle Swanson, the chair of the Board of Governors of the Academy. But another person who was very instrumental was Connie Herndon, who's seated here at the front. She's the Director of Strategic Alliances at ASM and played a major role in the planning of the Colloquium and the events that have followed the Colloquium. The conference that George mentioned, where we were competing with the Republican luminaries for space in that very busy hotel, was the next step. And amazingly to me, we didn't know what to expect. It was the first time ASM sponsored a conference on this subject. We attracted almost 400 international scientists to attend this conference. Hugely successful conference to talk about the impact of next-generation sequencing, not just in the diagnostics area of the microbial sciences, but in all of the microbial sciences. There was a major component of that meeting devoted to food microbiology and the use of next-generation sequencing to track outbreaks of food poisoning. So another very important event. The M.Bio article that George mentioned also, which was authored by the steering committee of the Colloquium with Brittany Goldberg, a colleague of mine at Children's National. Brittany is an infectious disease physician, pediatric infectious disease physician, and she's a terrific example of the beneficial presence of having young, early career scientists involved in ASM activities. And if you haven't seen the review article, which I know was at the registration table, pick up a copy of that to learn in a fairly concise format what the Colloquium was all about. Brittany did a fantastic job of serving as the lead author of that review. The coalitions. We have a coalition on next generation sequencing that again was the result of Marina and Connie putting their heads together along with me as the secretary to come up with the whole concept of the coalition in which ASM is staking a position, letting the world know that we are invested in this technology. We realize how important this technology is going to be to all of the microbial sciences, and there's no better society, professional society, than ASM to be at the helm of the employment of this technology throughout all of the microbial sciences. So that first coalition is well underway. The steering committee has met and planned the future of that coalition. Michelle Swanson and I served as the co-chairs of the steering committee, and now the coalition itself is beginning to gather to address all of the facets of next generation sequencing and what its impact will be in the area of the microbial sciences. Just to give you a little preview, we have another coalition that has just begun its formative stages, and this one will deal with antimicrobial resistance, which is not all that unrelated to the next generation sequencing because molecular diagnostics is crucial to advancing our ability to recognize antimicrobial resistance. There's no getting around that. And then we have a third coalition on the drawing board that will be dealing with the planetary microbiome, the human microbiome, the plant microbiome, the oceanic microbiome. Any microbiome you can think of we expect will be addressed by the third coalition that will be forming later this year. So a lot of great things happening at ASM in putting our society out there as being intensely interested and wanting to help shape the future of these important areas of the microbial sciences. Now, I just want to say a couple of words about what we're doing at Children's National Medical Center, sort of following in the footsteps of Charles. We established a pediatric genomics center in the Division of Laboratory Medicine at Children's. And so far, the primary focus has been in diagnosis of inherited diseases in children. So not microbial sequencing, but human sequencing. And this part of our laboratory has been in existence for about a year now, a year and a half, and has already made a tremendous impact on the health care of children in the Washington, D.C. area. We're readying a new area of the Genomics Center, which will be focused on microbial genomics. And again, Brittany Goldberg will be working with us in our laboratory to advance this project. And we hope to take advantage of some of many of the ideas that Charles described in his remarks. But of course, we'll be focused on pediatric infectious diseases, not only for diagnosis of infectious diseases, but you can use next generation sequencing technology to track outbreaks, just as the food microbiologists do. If we have a cluster of infections in the hospital that we suspect might be related, there's no better way than using detection of signature sequences from microbes to prove that that in fact is happening. And then ultimately, down the road of a couple of years at least, is the possibility of a laboratory like my own at Children's offering microbiome services to our patients. It's becoming ever more apparent that many health problems, human health problems, that we don't think of as infectious diseases have a microbial component. Our microbiomes that Stefano mentioned, where we are vastly outnumbered by the microbes in our body, it stands to reason they have a huge impact on human health, and they do. And so I foresee that diagnostic microbiology laboratories like my own will be in the midst of furnishing this type of service to our clinical colleagues to help them figure out what is going on with seriously ill children that might be traceable to the microbiome that these patients have. And so with that, I think I'll turn it back over to Stefano. Thank you very much.
Thank you, Joe. Thank you, everyone, for our distinguished panelists. I think it's abundantly clear at this point, I would say two things. One, from the science, that next-generation sequencing sits in this divide right now, where we've seen great success at the basic science level, at the microbiological level, at the genetic level, and there are boundless opportunities for the clinical side, but we're not there yet. So what does need to happen in order to reap the benefits of this powerful technology? So that is really the crux of the matter, and I think that could be a very interesting topic of discussion with all of you. But before we go there, I'd like to ask, and before we open up for a question from the floor, I'd like to ask our representative from the industry if they have any comments that they'd like to make on what they see important, what needs to happen for the technology to be adopted in the clinical labs, and how is the industry dealing with this? Mike, Kelly?
So, thank you for first involving us in this whole process. It's been an amazing experience, and it's interesting and appropriate that Charles starts out with his presentation, because I think I've sat in on at least 5 presentations that Charles has done, and every time he has this amazing story to tell, and when he gets done, instantly people come to us and say, I want to do what Charles is doing. Well, as you can say, he's obviously put a lot of work into this and developing pipelines and things, solutions to make it easier for others that come behind him. It's been actually interesting that George said that this document that we've all put together here is only 50 pages. When you put this many talented scientists in one room that have vast experience in microbiology, virology, parasitology, clinicians, scientists, and give them a lot of coffee, I'm surprised it's not longer. But I think that bringing all this talent into one room and talking about what their interests and experiences are has brought to light a lot of information that's very useful to not just clinical microbiologists, but the clinical market in general. And all of these concerns, I think they're all addressable things and items that are nice and clearly stated in this document. But together, we're going to need to come forward to address them. And certainly Charles has done a good job with pulling together bioinformatics solutions. But in a clinical market, that's not everything, right? And so groups like the FDA and HICA, who's working on a clinical microbiology database, NIST, who's putting together standards, these are all efforts that are coming forward individually with help from the scientific community, but there needs to be more. And I think that's really the goal of this publication, was to say these really are the needs, not only to pull the community together, but those that are interested in getting started in next-generation sequencing. I think clinical microbiology is really on the cusp of moving forward in this arena, and it's really exciting. Everybody wants to do exactly what Charles is doing. There's so many patients that die every single year of undiagnosed disease or illness. And we need to be changing that. We need to be solving these problems, and I think that we're making those steps in the right direction. I'm excited that Illumina is part of it. I think that the technology has evolved tremendously over the years, starting with the genome centers. I know George has been heavily involved in those efforts from a couple different genome centers over his career. And to see the technology evolve and improve in its accuracy and stability. But even so, as the technology has evolved, our systems have also evolved and become smaller. They're no longer these large standalone systems in the corner of a laboratory. Now they're accessible, they're on bench tops. And even today we have the latest system that's been launched, which is the mini-seq system, the least expensive system on the market, trying to bring this accessible to as many people as possible. It's just the first step. Education, training, all of these issues that we've highlighted, I think, cannot be addressed by one organization, but this organization is doing a great job of pulling the community together to address all of these things. And quite honestly, I'm really excited. I think we're really moving in the right direction. I'm excited to see the next steps that come along.
Thank you, Kelly. Thank you so much. Mike?
I think you can, or at least I realize how long I've known Joe. I had written down some comments, some speaking points, and I was thinking, how in the world did Joe get a hold of this last night? Because every last one of them he covered in his talk. As Joe said, I am a clinical microbiologist, and over the 30-plus years of my career, I've seen a handful of transformative technologies. EIA, automated blood culture systems, MALDI-TOF for the identification of microorganisms, PCR, and this. This is a transformative technology. Charles had mentioned that when you use polymerase chain amplification to identify causes of infectious diseases, you have to know ahead of time what you're looking for. The target has to be present in the mixture of organisms that you're assaying for. So you really have to have an understanding of where you want to be, whereas sequencing technology, metagenomics, and all the iterations are totally unbiased. You don't have to know what you're looking for. In fact, I feel that over the next 5 or 6 years, we're going to find things that we had never thought about. And one of them is the ability to distinguish people who are in a state of health from people who are in a state of risk for disease. And Joe mentioned evaluating the human microbiome as a measurement of the balance between health and disease. What is the responsibility of Kelly and Illumina and bioMérieux and any of the players on the industry side? I think, there was a saying at one point in time, I want to be like Mike. It was a Michael Jordan thing. Well, we could have a T-shirt that says, I want to do what Charles does. Be like Charles. And so the responsibility going down the road for industry is to make that possible for routine clinical microbiology laboratories. So to take this very technical approach to diagnostics, and I don't want to say dumb it down, but I want it to be simplified to the point where any clinical microbiology laboratory can put a sample in and get an answer out without having to have a great deal of technical knowledge. That's what's going to happen going down the road. Now, how do we do that? Well, we've done this fire drill before. We've done it with PCR. We've gone from something that only a handful of laboratories could do. I first learned the technology in 1991, and it was all laboratory-derived testing, and we got it to the point where any laboratory in the world, and even in developing countries, can perform these assays. So we have a blueprint. We can follow that. We can do the same thing with metagenomic analysis. But that's not as simple as it sounds. There's going to be a lot of innovation that goes into making this very easy for routine laboratories to perform. But I think in the end, It's just going to build on the information that we have on resistance mechanisms, on novel agents of disease, and how flora on the human body controls disease.
Okay, thank you, Mike. And now you'll know that the next report from ASM, the result of the coalition, will be a report titled, I Want to Be Like Charles, right? So we can, time to open up for question, and I would ask anyone who has questions or comments to please use the microphone in the back there and identify yourself.
Hi, Jean Jordan from George Washington University. My question is how to differentiate between association and causation when you have this unbiased metagenomics approach when we think back to Koch's postulate. Thanks.
Charles, maybe you can address that.
That's a great question, and it's actually a challenge that we face as infectious disease clinicians and microbiologists in that simply detecting the DNA or detecting even the organism within a clinical sample certainly doesn't mean that it's causing the disease. And this is exactly what we're facing right now with Zika virus, for instance, where we're detecting Zika virus in fetal brains, or it has been reported that's been detected in fetal brains, and yet we still don't know whether it's actually causing, or we haven't been able to confirm it causes the fetal microcephaly. So to answer your question, next-generation sequencing is really a tool. So it's a very powerful tool, and yet it still requires interpretation. And I view it simply as another tool in a clinician's armamentarium. So they would have to interpret the results in light of the patient's clinical presentation. So for instance, whether or not the infectious agent that was detected, whether that's clinically meaningful is going to be a matter of interpretation. I also feel that some of the tools that may be developed that may be along with next-generation sequencing is, for instance, the ability, one thing I didn't mention is that we not only have the ability to detect the organism, but we're also sequencing human DNA and human RNA. And because of that, we're also looking for the host response. And in principle, we might be able to mine that data to be able to determine whether or not the organism is actually causing an infection or is simply colonizing that body site. So, and I think that in the future, this might be a way that we can take full advantage of all of the data that's the sequence data that's being generated from next generation sequencing.
Thank you, Charles. Any other comments from the panelists?
Yeah, let me just maybe come at another direction at the question too. We did some studies of samples that had gone to a clinical lab from patients who had diarrhea. We did metagenomic analysis. And the main agents that had been identified by the clinical lab, C. diff, things like that. In I think 10 or 15% of the samples, though, we found a second plausible agent, Campylobacter, Salmonella, that had not been reported from the diagnostic lab because that may not have been the one that they were looking for, or something like that. So, what's causative? I mean, I think in the world of trying to make a decision about a therapy or something, it's more a question, I think, of you want as much information as you can get in order to see the whole landscape of what's there. I don't know if you want to add something to that, Charles.
Any other comments?
Yeah, one other point I would like to make, as informative and exciting as next-generation sequencing is, and molecular diagnostics as made possible by PCR and other amplification technologies, the detection of a gene that may have potential virulence production in a patient is no longer enough. Yes, that information is interesting, but I foresee that because we don't know that a detected gene is actually being expressed, detecting the DNA is one thing, but is that DNA being transcribed into messenger RNA and being translated into protein is another very different question. And so I foresee the combination of next generation sequencing, other molecular modalities with proteomics really becoming the centerpiece of of infectious disease diagnostics and the recognition of antimicrobial resistance markers and other virulence factor markers as this ability becomes possible for us to employ, we're going to want to be sure that what we're detecting is actually clinically significant. So I think proteomics is something that we're going to have to see. I'd be interested to hear what Charles has to say about the combination of a proteomic and a molecular approach toward diagnosing health problems.
Great. Yeah, so I completely agree. I mean, I believe that proteomics is probably kind of the next step. And I think that what you'll see in the future is basically integrating kind of several technologies together in terms of making interpretation. In addition to that, I do also want to make the point, although ultimately what we want to do is we want to have kind of a sample to answer pipeline and have a seamless answer. I think in reality, because of the complexity of the data and exactly as you brought up in the question about the complexity of the interpretation, that at the beginning that it's certainly possible, I think it certainly would be more practical to have the data interpreted, say, by a pathologist. So this would be, for instance, it would be akin to for instance, pathologists that are interpreting, slides, for instance, histological slides, or even a radiologist interpreting a chest X-ray or a CT scan. That in a similar way, there may be a genomic pathologist who's trained who will be able to interpret and really be able to consolidate all of the data that's being produced from the next generation sequencing profile.
Yep. Any other comments here? No? Okay, next question.
Hello, I'm Heike Sichtig. I'm at the FDA, and I was also part of the steering committee. And I really would like to hear from you guys, based on the experiences that we've been gaining from these events, from laboratory experiences. What would you say to a patient at the end? I mean, because we're doing all this at the end for the patient, and if there was one take-home message that you give to the patients, what would it be in terms of microbial NGS?
So patient perspectives, who would like to take it?
Everyone's looking.
Jim from...
Well, from my standpoint, the information has to be clinically applicable. The information that's provided has to lead to potential therapy. Something that is actionable, whether it be a change in antimicrobial resistance, whether it be the possibility of an autonomous or autologous or a artificial transplant in the gut, but something that has the potential to benefit the patient. In that sense, we're going to have to start looking at things entirely differently. With the advent of stool transplants and a number of companies developing artificial microbiomes as a drug, we're going to have to look at things from an open-ended standpoint. So, does this benefit in the long run? And to do that, we're going to have to take a leap of faith here. We're going to have to start using these technologies in the RUO, research use only side, as a means of developing a in vitro diagnostic test, instead of kind of the other way around, where we tried to develop an in vitro diagnostic test and then some aspects of it are left on the RUO side. So we're going to have to do a number of preclinical evaluations, but to be allowed to do that, to be sanctioned to do these studies to get an end result.
Yeah, I would like to add to Mike's comment. I think one of the most important challenges that we in the microbial sciences have, especially with respect to this new technology, is educating the masses as to what this is all about. What is the clinical significance to the patient of a laboratory being able to detect a gene or detect expression of a gene? And I see the ASM having some responsibility for that educational process. We pride ourselves on the resources of information we have within our membership, and that's wonderful. But another goal I think that we need to have is to explain this new technology to individuals outside of the American Society for Microbiology of what does this mean? What does this mean to the patient? Heike asked a very good question. In healthcare today, at Children's National, our CEO stresses that we are a patient-centered healthcare environment. We bring services and help to the patient. We don't send the patient wandering around the hospital looking for help. We want to bring all of our experts to the patient. And I think the laboratory has a responsibility in that area too, of being able to explain to the patient in language that's understandable what is this technology all about and why is it important to me as a patient. I think that's a challenge that Stefano and I have talked about since he's come on board as our CEO at ASM. And we have an excellent education department at the society headquarters that I think will be playing a role in this as we move forward.
And on this, I'd like to kick the tires a little bit and ask George a question related. The report talks about an issue which is, I think it's very relevant to this, and it is how to transmit information coming out of next generation sequences to the ID doctors or others, in making sure that the data is interpreted correctly, because we don't control at a certain point how the information will be used, and it can create confusion, anxiety, and problems in the delivery of care. So, George, would you like to, if you think it's appropriate at this point, maybe mention recommendations, or how do you think, or how the committee thought this could be handled, and if there's anything that we can do in terms of education guidelines to ensure that from the information to what the patients really need to know and answer is done as seamlessly as possible.
Sure. So I want to also make another comment afterwards about the other question that Heike had asked. But I think one thing that the members of the committee were sort of the fear is that when you do a metagenomic analysis, you produce so much information that you have a danger of not just swamping somebody with information, but providing so much information that's sort of irrelevant, but could be distracting. And so a big challenge with these data sets is to try to figure out how to analyze them in such a way that you kind of get to what's at the heart of the matter. And I don't know what the answer to that is. I think everybody agrees that, maybe you have to do clinical trials, maybe you have to do a lot of things and try to figure out, now in a diagnostic lab, you might be testing for the most common pathogens or something like that. And that's going to cover 70% of the cases. And then there's 30% of the cases where it's just not on your radar screen. Or if you're in a bone marrow transplant unit where patients are immunosuppressed and all kinds of things are growing in them, and you don't know what's relevant for an infection or not, or things like that. I think there's a sort of medical knowledge question here, too, about how to apply experience, almost empirical knowledge about what people have seen and what people have learned in the past to take these enormous data sets and make them into something that gets condensed down to now maybe getting the 70 most common pathogens. Now maybe we can get up to the 90% or 95% of the most relevant case, decisions that need to be made. I think that's kind of the spirit that I would look at it. I don't know, Charles, you want to add to that? Sure.
Yeah, I just want to make one comment, is that as part of our prospective study of next-generation sequencing at University of California, San Francisco Hospital, we will be implementing an infectious diseases precision medicine board, and it's in a similar model as tumor boards for treatment of cancer, where we want to take advantage of kind of individuals with various levels of expertise in genomics, in microbiology, in infectious diseases, along with the the treating clinician who actually ordered the test or is planning to order the test. And in that way, I think that we will go over every single case, and we will and then follow outcomes and also follow recommendations and make a joint recommendation for kind of these cases, at least for this prospective study. And I think that by doing so is that we'll be able to arrive at a consensus and have a better idea in terms of the indications and when should we do this test, when is it appropriate to do this test, and also have a better idea of how to interpret the test in a way that is best for the patient. I also think that even prior to educating patients about this, we really need to educate clinicians, and because the clinicians ultimately are going to be the infectious disease doctors, the other clinicians, surgeons, hospitalists. These are the clinicians are going to be the individuals who will actually be educating the patients about the tests. So first, we really have to educate clinicians about the technology and about the test and how and what are the indications for the test.
Yeah, I think this is a great point, and what's emerging is really, I see the issue of bioinformatics and data analysis, which in a setting like Charles, in an academic, it's easier, but in a standard laboratory poses a big challenge. In a sense, is what we saw with SNPs, right? So we said a lot of SNPs, and that's a risk factor, but what does it really mean? And that's where the interpretation becomes very challenging. So I think this is going to be something very important that ASM will tackle. I think George has a question, and then
to go back to
you had another point.
To Heike's question. So most of what we're talking about are pathogens and looking for things like that. There are going to be other microbiology-related applications of this that it's also worth keeping in mind in the sake of this discussion that are relevant for what the patient needs to know. We did a lot of work in the neonatal intensive care unit and with infants, and there what you're looking at is you're trying to get the picture, what's the proper colonization of the GI tract for a newborn baby? What things are there that might put them at risk for necrotizing enterocolitis, for a bacteremia, for things like that? And in those situations, you can almost do real-time monitoring of what's going on. Sampling is non-invasive. You just get the diapers, you get a stool sample. You can do it daily or however often that happens. You can do these rapid microbiome analyses and metagenome. And somehow you imagine that there will develop a picture of what's the proper colonization, what should be there at any particular stage in the development of the baby, and what things stand out as not being quite right, or somebody who's starting to overgrow, or things like that. That's all other kinds of interesting information that I think can inform how better to deal with the development of babies, and it's all based on the microbiome. There's all kinds of stuff in the literature about autism having to do with the microbiome, or other kinds of things that you would think might have a more indirect relation to it. But for all we know, there's things that happen in that early development of the microbiome that can set a baby up for some downstream medical condition. And I think we'll start to find that out as we can apply cheaper, faster, comprehensive analyses of the colonization of a new human with the microbiome too. So other kinds of information to be thinking about.
Thank you, George. So we have Kelly, and then we have two more questions. And we have 10 minutes, I think. So I want to make sure that we get everything done.
No problem. So I just wanted to revisit Heike's question. Heike, apparently this was a really relevant question for all of us, but I think not only from the microbiome's perspective, but the public health perspective, this is really important. As we're all living longer, we're seeing pathogens, we're interacting with the flu, everything else. As a parent, we run into situations where our child spikes a fever and we wonder, do we need to run and get an antibiotic? Do we need to go to the emergency room? What kind of time frame or window do I have here? You hear patients, parents whose child passes away and it was just a fever for 24 hours and they're passed away. These are all experiences that none of us want to undertake, right? But I think a lot of it that needs to be addressed is even at the insurance level and the reimbursement to the microbiology labs. This is one we haven't really talked about. We highlighted it in the article, but there are a lot of patients out there that are honestly sick, legitimately sick, and are not able to get the care that they need because of what their insurance is willing to pay, and that they can't afford to pay outside of that. So these are all big concerns that clearly we cannot address at this table, but they're things that need to be talked about. How do we figure this out and solve it so that it does affect patient care?
I think the issue of reimbursement is a really big one, and that's something that we definitely need to deal and facilitate, and working with you and the federal agencies will be very important. Thank you, Calvin, for that perspective.
Michelle Swanson from University of Michigan and from the Academy. Charles, I'm curious, in your group's experience with patient samples, what % of the runs, the reads, don't match? So those could indicate that those orphan sequences might indicate gaps in our technology, the computational biology, or how little we really know about the trillions of microbes that we live with us or that we encounter in animals, food, water, and soil.
Thanks for that question. So the question is in terms of the deep sequencing reads, how many of them don't match to any organism, so the answer to that question, it depends on the sample type. So when we think of more environmental samples, and these are stool samples or respiratory samples that are in direct communication with the environment, typically a large fraction of them may not match. For instance, in stool, up to 50% of reads, actually sequence reads, don't match anything in the database. And I think part of that is that it sort of reflects sort of like the gap that we have of not only the human microbiome, but also the environmental microbiome. Certainly these kinds of specimens are in direct interface with the environment. For more sterile specimen types, say if you look at blood, for instance, serum, or if you look at cerebrospinal fluid, we are able to actually identify the vast majority of sequences. So in terms of how many remaining that do not match, we're talking typically, in some cases less than 5%, or in some cases even less than 1% may not match. So, and I think that is also part of the reason why we've chosen to focus, at least initially, on cell-free or more sterile fluids, such as cerebrospinal fluid or plasma, for infectious disease diagnosis, because the interpretation is definitely more straightforward and easier. For these other types of specimen types and other types of diseases, like such as respiratory secretions for diagnosis of pneumonia, this will be more challenging, and part of it will be sort of interpreting. It gets back into the question of if you look at a complex microbiome, I mean, which combination of pathogens or which microbe is the pathogen is a key question there. And I think that is where we will certainly need to provide or have some clinical interpretation, really, for it to be used to make an impact.
Thank you. You're welcome. Next question.
Emil Esho with the DoD. One question for Dr. Chiu, and then one quick question for the group. Patients usually who are that sick for that long, and I think you alluded to, usually receive many empiric antibiotics, many of which would have been effective against leptospirosis. Well, did that patient receive those antibiotics, and if so, why do you think he didn't respond to that? And then for the group, the Antibiotic Resistance Leadership Group recently published a report on rapid molecular diagnostics for AMR, and a whole genome approach, curiously, was not included in the 4 assays the 4 techniques that they reported on. Can you conjecture or comment on why you think they didn't include a whole genome approach?
Okay, antimicrobial is a topic.
Yeah, so I'll just answer the first question about the neuroleptospirosis case. So it is detailed in the paper somewhat, but the key question was when the patient presented, actually the thinking was that he had an autoimmune disease, because he had actually presented with numerous autoimmune manifestations. He had a conjunctivitis. He developed a condition called idiopathic thrombocytopenia, and these are typically autoimmune manifestations. So the suspicion was very strong, at least when he was seen by his clinicians, that he had an autoimmune disease. And accordingly, he did not actually receive empiric antibiotics until sort of relatively late in his course, basically by his third hospitalization. He had actually gotten, interestingly enough, he had gotten antibiotics inappropriately because he was given antibiotics because he had had an extraventricular, a neurosurgical device that was placed in his head to decrease the pressure in his head. And they had actually inappropriately put him on those antibiotics for prophylaxis against the neurosurgical shunt that he had. It turns out that was actually cefuroxime, which has activity, but it does not have good activity against leptospirosis. He was not put onto broad-spectrum antibiotics until very late when he was already in intensive care unit at that time. And he was put on a regimen, at least initially, the regimen that he was put on, he did not respond to. And it was over that weekend that we were then able to identify that he really had a case of neuroleptospirosis that was simply given sort of ancillary treatment that was not targeted against that organism.
Any other comments on the issue of antimicrobial resistance that was mentioned?
Yeah. Well, I can offer a comment. I think next-generation sequencing or even PCR is extremely useful for recognizing known antimicrobial resistance markers. But recognizing unknown antimicrobial resistance markers is another challenge that we may be able to overcome it, I think, with sequencing if we can look for patterns of sequences that are predictive of possible resistance. But I want to go back to the patient-centered question. So the patient who is in the intensive care unit ill, like the patient Charles described, who may have an infectious disease, what that patient needs to his or her clinicians to know is, what antibiotic is my organism susceptible to, not resistant to? And it's going to be a long time, I think, before we can employ molecular technology to predict susceptibility. And so I think we're still stuck with phenotypic rather than genotypic assays. And the phenotypic assays today are growth dependent. I think we're going to still be depending on, for a good time in the future, exposing the organism from the patient to various antibiotics and seeing what the in vivo effect on a culture plate is of exposure to those antibiotics. That's what the patient wants to know. That's what the patient's clinicians want to know. So I think we're going to continue to see microbiology laboratories relying on culture-based technology along with the molecular technologies and the proteomic technologies that we described that are just around the corner. It's going to be a while, I think, before we are not doing growth-dependent susceptibility testing assays.
Yeah, just to,
Mike.
Just to add to that, thank you. You've made mention to the longer application of some of the technologies that our company produces. But you're right, we can't predict susceptibility from sequencing at the moment. We will with expanded database collection over time where we can get pretty accurate. But I think this will all be followed up with phenotypic susceptibility testing where you would issue a report on drugs to avoid in this particular case because you've detected genes of known resistance, but you won't issue a report of susceptibility until the growth-based method comes out. Same thing holds true with direct detection of organisms from blood. We just don't have that level of sensitivity yet, so it's likely that we will continue to back up direct detection methods with blood culture for a very long time.
Thank you. We have time for one more question, if there is one. If not, I'll ask one thing related to this, which is the report touches upon, and I don't know, George, if you want to take this, but or anyone else, is about data mobilization. I think it's connected to what Mike just said. So do you envision a federally based database like NCBI based for data in a secure way, HIPAA compliant, to be able to do research out of the information that is gathered out of this, which could be very relevant to detect susceptibility and resistance. Any thoughts on this topic? Seems like something important for the future to think about.
I think there are initiatives underway. I don't know if Heike wants to talk about any of the things, but I think that certainly is the intent, that there are already initiatives. I know Mike's company had thought about trying to do something and realized that there's so much stuff going on in the public area right now that it's better to chime in and support that rather than have private ones that could then be licensed or things. So there's a lot of action in this area, but it's a hard problem to have comprehensive, relevant, high quality, highly curated databases to be able to satisfy.
Absolutely, and what you're
this.
hearing here is a great interest in ASM to be a facilitator in this because it's so important, both from a scientific and clinical aspect, that anything that we can do to facilitate this, working with the agencies and the companies, we're here. Kelly, I think you have
so along that effort, Illumina has an offering called BaseSpace, and this is a cloud-based software analysis shareware tool that allows people to collaborate internationally. You can push data to share, or you can analyze it in the cloud. I don't know if I mentioned it's hosted by Amazon. But this, by the end of this quarter, will be HIPAA compliant. And the idea is, I think I've talked to Heike about this many times, is that the database that they house needs to be accurate. The data needs to be of good quality. I think there's groups like NCBI now that are coming back to their databases and saying, how do we mine this database for accuracy and good data,
QC.
Curation, annotation, whatever from there. And so the idea with BaseSpace is that you have a forum for analysis tools that then can be pushed up into with an SRA submission or whatever tool that is developed from the FDA on the Argos side, that we could push data directly from that HIPAA compliant database into once it's been deemed good quality by the user, then could be pushed into a database from there.
Excellent. Heike?
Thank you, George, for bringing it up. And I would like to just mention that FDA is spearheading an effort to create a clinical reference database for microbial genomes. And this has been an effort ongoing for a few years now, and it keeps ongoing, and we have funding to populate databases. The database is called FDA Argos, and it's hosted at NCBI. So in terms of sustainability, it will keep going on. Even like with many projects where funding runs out, this will be still available, and we will qualify genomes that are already existing into the database. So there's no really stop to this database. And I would like to take this opportunity as a plug for asking if somebody has samples and we have gaps. And Chelsea Gaia also works at FDA with us on this database. So you can definitely contact us if you want to contribute. We are open for any collaboration. And ASM has been a great help for really putting this forward and for publicizing it and for helping us out with sample acquisitions. And I also want to thank Joe Campos and Brittany for really spearheading this as well in terms of clinical samples from Children's National Hospital. It's been very helpful.
Thank you very much, Heike. That was a very important point. So we have come to the end of our program. I think we could discuss at least 100 more topics. High throughput discussion could be. But unfortunately, our time is up. I would like to conclude by, first of all, thanking all of you for being here and finding time to listen about this very exciting topic and painting the picture of how the future is going to be in a clinical microbiology lab. I'd like to thank our distinguished panelists. Thank you for your presentation. It was very illuminating. And I'm not referring to any particular company. And I would also like to thank the Academy, Michelle Swanson, Marina, the Director, the Strategic Alliances Department, and all the staff at ASM, in the Academy and at ASM, that have been so fundamental to get everything done. So thank you, and stay tuned for next steps with the coalition and any other initiatives that we may have. Thank you again.